Hello and welcome to the last Beyond Biotech podcast of 2022, and it's the 27th podcast. I'm Jim Cornell from Labiotech, and this last week before the holidays in many parts of the world has been a whole lot busier than I expected. No sign of the news drying up. I've been busily posting videos that I finally got to edit, interviews, and a whole lot more. And when you throw in wrapping gifts, last-minute shopping, having to take the car in for repairs and sick family members, it's been kinda crazy. And on Sunday, after all the gifts are opened, my job title changes from Editor-in-Chief to Assistant Lego Piece Finder, but it will be fun. We're also in the middle of strikes taking place here in the UK with nurses, ambulance drivers, postal workers, teachers and train drivers among those looking for better pay to keep up with inflation. It's certainly been a very interesting year. I appreciate that as this is the last podcast of the year, it comes at the busiest time of the year, so it will have a special gift this week. And that gift is less of me, because it's a bit shorter. I figured with everyone travelling or shopping that a shorter one would be more appropriate, so this week there are just 2 interviews, but both extremely interesting ones. We have conversations with Talat Imran, CEO of Rani Therapeutics, and Stef van Grieken, CEO and co-founder of Cradle. So now it's time for the news you may have missed over at Biotech.eu. SoftOx published its chronic wound study report, Lift Biosciences received a double boost, and Sanofi and Innate Pharma expanded their natural killer cell deal. Urogen completed enrollment for its bladder cancer phase 3 trial, and I also posted a whole lot of video interviews from some events that I attended. Green Bioactives is growing medicines in plant cells. F2G is looking for FDA new drug application approval for its invasive fungal infections treatment. And Ribbon Biolabs said a recent milestone will lead to entering the market in 2023. Verona Pharma's drug has met its COPD Phase 3 trial endpoints. The approval of Ebvallo ushers in off-the-shelf T-cell immunotherapies, and research shows artificial DNA can kill cancer. Stem cell plasters could revolutionise heart surgeries. Anoka secured €25 million of funding to develop next-generation T-cell therapies for cancer, and Poolbeg identified new potential drug candidates for RSV infections. Biomica raised $20 million to advance its microbiome-based therapeutics pipeline. The Spanish food tech sector grew in 2022, and 4 Japanese life science companies are looking to reduce their environmental impact. Ganymede Bio raised $12.75 million for its data platform. The world's first CAR-T technology trial for patients with T-cell leukemia subtype has been authorized. And we had an article on 10 biotech companies to watch out for in 2023. And you can read all of these and a whole lot more at Labiotech.eu. And that brings us to the first of this week's interviews, which is with the company Cradle, which recently exited stealth. The company has raised funds to help researchers design and program proteins. To tell us what it means is the company's CEO, Stef van Grieken.
Cradle is a biotech startup. We've built a design platform for protein engineering that helps teams in pharma, food, materials to engineer better proteins with help of large language models. It's a little bit akin to ChatGPT for proteins, and so we help you design better proteins. And really, the goal is to make it a lot easier to do protein engineering for these types of companies. So hopefully, over time, it becomes easier to replace things like milk by using microbes, make better medicine, and all of these lovely things that you can build with biology, but then for a lot less R&D budget.
Could you tell me how the AI portion of this works and the cell factory concept?
Yeah, sure. So we're using generative machine learning models to design proteins and protein functions. And the way that they work, if I were to use an analogy to natural language, is I don't speak Swedish, for example, but you could imagine I can show you maybe 1 million pages of Swedish. And then I could show you a next page of Swedish, and you could say 98% Swedish, these parts don't look very Swedish. If you translate that into, let's say, DNA or the world of proteins, you could imagine that you can learn, hey, this sequence looks looks okay, looks very natural, or something that nature could have come up with. And these are some of the parts that look maybe not very good. And so that's how we avoid, let's say, making a lot of changes to proteins that nature probably didn't intend. Then the second thing you can do is sort of condition it towards something that you want to improve. So for example, protein engineers might want to improve the stability of a protein so it can survive under different temperatures, or the binding affinity so that it attaches to a molecule or another protein much more effectively. And we can sort of what we would call condition the model. So again, I could show you a bunch of additional pages with, in the case of stability, the temperature. And then you could maybe say, this looks like very warm protein, but these parts make it look not very warm. And so that gives us a really good sense of where can we make modifications that might help. And then finally, we use experimental lab results that come back from people's labs, what we call fine-tune or improve the models as they learn. And typically they would sort of start accounting for, the assay that people use, some of the context in which the protein tries to perform. And I think one of the things that we've shown, at least for thermostability, is that these types of machine learning models perform much better than, let's say, conventional homology or energy-based simulators, or some of the tools that people are using today. Yeah, and we're basically building a platform, like a web-based application and a set of APIs that people can then use to easily implement that and use those types of tools within their own research and development departments.
As far as the proteins are concerned, what's the initial building blocks for those? I mean, as an example, you mentioned milk. Would milk that's created in this way be vegan, or would you still need the animal product in the beginning?
To reframe the question a little bit, but typically machine learning models work well on properties. Like, for example, AlphaFold, works on many different structures, not as good on all of them, but typically, for different domains, AlphaFold is a useful thing. Similarly, what we found for example, protein stability, it works across different types of use cases. So whether it's, let's say, the stability of an antibody, making sure that it stays stable within a human body, for long enough so it can find a target, or having a stable enzyme in some metabolic pathway. Those are typically very similar problems for the machine learning models to solve. A set of the types of users that we're trying to target is in the alternative protein space. And so there's companies like Perfect Day that are making milk protein using a microbe, or filamentous fungus, I believe. And you can imagine lots of protein engineering problems that a company like that might have, such as, I would like to express more of this protein, or I would like it to secrete better, or I would like to be more stable. And so typically we would start from a natural protein or something that can be found in nature, and then help teams evolve it to have better properties for the product that they're designing. But we aren't like making a product. We're like picks and shovels, like Figma for protein engineering. We're not, let's say, married to one class of protein or a specific product, if you will. And then would you still call a milk product vegan? That's a good question. You should probably ask Perfect Day. And it's an open question. A lot of the companies we talk to in the alternative protein space, they actually have these questions as well. And for example, regulators are not really giving them a lot of good guidance. So for example, can you do recombination of known like bovine proteins? Is that still natural, or do you need to actually show that there's an organism that produced it in exactly the same way? Open questions, but wide open frontier, I guess, and a good conversation to have had both with.
And then there's also not just regulations, but there's also labeling regulations. Is it, if you create something that's a vegan product of meat that's a replica of meat, are you allowed to call it meat? You get into, and I know that's been a huge issue in the US.
And it's only going to get more complicated with the hybrids, right, where you have both plant and animal ingredients, right, in the formulation. And anyway, bold new world.
Yeah, for sure. Absolutely. So what kind of applications are there for your technology? It seems like it's kind of sky's the limit, really.
So machine learning typically works on different tasks, right? And so, we've seen last year that AlphaFold made a huge breakthrough in the prediction of a structure of a protein. And I think that's helped a lot of research organizations. I think the next step is a lot of protein function prediction and generation. And so what we're focusing on is things like the stability of a protein, the activity of a protein. Will it express well? Will it bind well? Will it be specific? So only bind to a very specific target. And I think, to your point, there's a lot of people that are using protein engineering techniques today across a wide range of different applications. I do think that by lowering that barrier, more people will start doing it. I would say typically, if you're in pharma and you're making a biological, like a CAR T or an antibody, that's an area where this kind of technology can be very useful. If you're a company that's sort of doing biocatalysis and you have some enzymatic process to make a chemical, that's definitely an area where we can help. In the alternative protein space, we're seeing a lot of people that are trying to improve secretion or expression of a certain protein. That's definitely an area that we can help. But I think, to your point, it's a little bit like you can do a lot of things with Figma and you can design a lot of things. It really depends on whether there's a specific tool that we have in our toolkit that will help you right now. So broad range of applications. Definitely. We are focusing right now on expression and stability as sort of the first two design tasks, given that we're still a young company. And so, most of the design partnerships that we have and some of the early users that we have are working on those types of problems.
Long term, how will this kind of technology help to reduce the time it takes for things to come to market and also the cost?
Yeah, for sure. I think the general remark I would make is in biology, we've become very good at increasing the number of experiments that we can run in a unit of time. Like throughput has definitely gone up. And therefore, we've basically been able to get lucky more often, right? So we have more shots on goal, if you will. I think a founder that we interviewed of a smaller biotech company put it really well when he said, 99% of my engineered proteins don't hit their design specifications. I just don't know which 99%. But I do know it's about 80% of my runway, right? Because it costs $150 to try one. And so I think really where generally generative machine learning can help is really improve the number of shots on goal that you actually hit. And so therefore, the relative proportion of proteins that you try in your lab have a higher chance of succeeding. And so how big that will be will probably depend on what market you're in and what you're targeting. But even if we could bring this particular example of this founder from 99% to 95%, that's like a 4x improvement in your success rate, which means you can learn faster, which means you probably will get to market dramatically faster. For us, the goal would be to like one order of magnitude improve the chances that you hit your design targets. And therefore, your cost for the part that is protein engineering in your product should go down by a similar order of magnitude. I think more broadly, there's often the discussion, will machine learning solve, and then you get a big banner title of like drug discovery. And I don't think that's the right way to think about machine learning. Machine learning is usually very good at a specific task, right? And so in drug discovery, before AlphaFold, doing a structural analysis of your product required an X-ray, right? And that was a very expensive thing to do. And now with the coming of AlphaFold, that cost is dramatically reduced for that task. I think very similarly, for example, in drug development, we'll see more lead discovery and lead optimization tasks. Those will start to get a lot cheaper. My prediction is in the next coming years. But there's still a lot of things that are very complicated, like, is this safe to use in certain, patient classes? Those are very hard questions to answer. And it's unclear at what point machine learning will make a big dent. I do think, like, for example, in our company, we will never claim that at the cellular level, we're able to do anything useful. We just don't think the machine learning is there right now. But at the protein level, we're definitely starting to see a lot of very good developments. And I think that's definitely a domain that's going to be disrupted in the near term. And so it will still, for a lot of things, require the good old ways, but increasingly parts of your R&D programs will start to become a lot cheaper.
And kind of a bit of an amorphous question, but I wonder if you could give me a bit of an overview of where the synthetic biology market is at right now and where it could go from here.
Yeah, for sure. I think it's still in its very early innings, although it's been around for a little bit. Actually, very similar to machine learning. I was very early at Google in some of the innovation that was happening in machine learning. And at the time, the joke was made that in ML in the last 40 years, you could basically give approximately the same presentation at a conference up until ImageNet, sort of in 2004. My sense is that synthetic biology is very much at that same tipping point, and it's driven by cost of sequencing is coming down really fast. And so reading whether you've done something correctly is cheaper. Assays are becoming cheaper. Making DNA is becoming cheaper, like some of the enzymatic DNA synthesis that are methods that are coming out, where it's like an order of magnitude cheaper to try something out. I think is going to drive a lot of that. And I think ultimately the output market is massive, right? If you, I think McKinsey calculated that about 60% of our like raw economic inputs that we consume, whether it's food or clothing, like I'm wearing a nylon t-shirt, or chemicals or medicines, or, all of these things can slowly be replaced by biology. That's just a super fascinating future. I think one of the things that I am at least starting to see is more sort of horizontalization of different components that you need in the stack. So where if I look at, for example, Zymergen, when they started, you had to like literally build everything from the ground up, right? So there was no limb system, there were no robotics automation companies, there were no components that you could sort of take and then build your company on top of that. Increasingly, I think we're seeing that, like with a company like Benchling that helps you run your limbs, companies like Twist and IDT that can sort of provide you with the right sequences, companies like ours that can help you in designing proteins, and sort of increasingly, I think, quote unquote, the stack is there to make it dramatically cheaper. I wasn't there in the 90s, but to some extent, biology feels a lot like computing in the 90s, right? If you wanted to ship a product to everybody globally, you needed like data centers, you had to build all of these things yourself. And increasingly, you don't have to do that. So you can sort of focus on what's really the core product that I need to build, what are the core essays around that product, and everything else I can sort of not build myself, but just rent or hire.
You recently received some funding. I wonder if you could tell me what that means to the company.
Yeah, sure. So we were founded in 2021, end of last year, and we now just emerged from stealth a couple of weeks ago. We raised 5.5 million euros in seed funding in a round led by Index Ventures and Kindred Capital, as well as some really awesome angels like Emily Leproust from Twist. Yeah, we plan to basically use this funding to help grow the business as we bring on new design partners. So today, there's a handful of people that are using the platform, and we're trying to scale that out to the next 10 or 15. And so really looking to connect with more companies out there that have stability problems, that want to engineer proteins with better stability properties, that want to improve expression or want to use structure prediction. And hopefully with that next, let's say 10 to 15 over the next year, we can be ready to sort of release the product sort of in a more open fashion to the market. I think and you might have seen this with technologies like ChatGPT, like you kind of need to really make sure that the quality of your predictions is good. And you've probably seen a lot of those examples where people are like, haha, if I type in this prompt, it's saying something ridiculous. We kind of want to make sure that we can guarantee that if you use our software, it actually produces better proteins and not something that looks like a shiny protein. And so a lot of the quality work is what we're focusing on this year. And I think, especially with scientists, rightfully so, we need to show, look, it works for most of these, and you can trust us to use this type of software. So that's what we're focused on in the next year.
Where do you expect to be at kind of a year from now, I guess? What's the roadmap?
Yeah, so a year from now, hopefully we can launch the technology sort of at full scale and start onboarding more people in a way that they can just use it without our help. That's where I would like to be in a year from now. We'd like to start adding more protein engineering design tasks. And so, we talked a little bit about binding affinity. We'd love to also work on activity for enzymes. We'd love to work on specificity and those types of tasks. We'll need to develop models for, we'll need to develop data sets for to train on, and we'll need to make those available to our customers through the platform. Yeah, and then, hopefully in a year from now, we can start shipping that as software, make it available to customers. We'll always make it available free of charge to academia. So working with a bunch of academic partners as well, growing the team a little bit, and ultimately getting into a world where any team can do protein engineering relatively easily and get to, proteins with the right design specifications, in a matter of months and not years.
How are you finding those partners or people to work with?
We've actually been quite lucky. So we came out of Stealth at Slush, which is a startup conference, and there was a little bit of mention of our out of stealth in popular media. We've got over more than 200 companies and academic groups that reached out and wanting to try sort of our early software. So that's been really encouraging and exciting compared to I'm from classical tech, right? My previous job was on the product leadership team of Google Research. Things with software are a little bit slower in bio, and it sometimes takes a little bit longer to get to a deal. But, that's part of being in this sector. I'm super excited about the future. I wrote a little article on how more people from proper tech should go into biotech, because it's probably one of the new awesome frontiers that people with that kind of talent should go work on.
Next, it's to another really interesting subject, and that's a pill called the Rani pill that is designed to do away with injections. It's a pretty nifty piece of technology, and to tell us all about it is Rani Therapeutics CEO Talat Imran.
Absolutely. Rani Therapeutics is a clinical stage biotech company. We're taking injectable biologics and turning them into pills. This has been something that has been tried and attempted since basically the 1920s when insulin was first discovered and started to be used. But really in earnest over the last 40 or 50 years, there have been dozens and dozens of attempts, primarily with the smallest of peptides, to turn them into an oral formulation. The reason it's so difficult is all biologically derived or inspired compounds are organic, and our gut views them as nutrition and destroys them before they can get absorbed. So the attempts that preceded us, we call them chemistry-based approaches, were where you'd add excipients, additives to the drug to try to enhance absorption, mucus denuders, permeation enhancers, enzyme blockers, et cetera, et cetera. And the net of it is that you get very, very poor absorption, little to no absorption, maybe bioavailability of 1% or less with extremely high variability. Not to mention that doesn't work for larger, like monoclonal antibodies or large proteins. We've taken a completely orthogonal approach at Rani Therapeutics and developed what's essentially a swallowable auto-injector. It looks like a regular capsule. It has a pH-sensitive coating on it that protects it while it's in the stomach, in the acidic environment of the stomach. It exits into the small intestines. There's a self-inflating balloon that'll adjust, that expands and aligns the syringe that's inside of that balloon. And that syringe injects, or deposits, I should say, a microneedle in the intestinal wall. There are no sharp pain receptors in the gut, so this is a pain-free injection. The balloon itself deflates and is excreted out, and the needle dissolves, and there's a drug, a microtablet inside of that needle that gets absorbed. So this is, like I said, essentially a swallowable autoinjector, and we're getting absorption rates that rival a subcutaneous injection, regardless of what's put into the Rani pen.
Sounds interesting. What are the issues with the current methods of drug delivery?
Well, at its most basic form, people hate injecting themselves. There's a real aversion to needles. And naturally so. We don't, in any kind of recreational sense, poke ourselves with sharp objects, right? The needles have gotten better, and a lot of them have very little pain associated. But there's, like I said, there's just this natural aversion to injecting. And so the problems that come from that are not obvious. The obvious thing is there are patients who won't take these drugs. Clinicians are less apt to prescribe these drugs, even though biologics are highly effective earlier in the care continuum. So patients tend to be so sick that they're willing to take these drugs. But then the way they're formulated is also a problem. Pharma companies know that patients hate injecting themselves, so they formulate the dosing for infrequent injection. So maybe once a month, once every 3 months, once every 2 months, whatever the case is. And that leads to high variability from the peak to the trough in terms of serum concentration. And that can lead to either adverse events or limited efficacy or reduced efficacy. So getting to a daily dose oral administration could solve all of these problems.
And you kind of explained the Rani pill. Is that how it works? It goes into the intestine?
Yeah, it goes into the intestines and deposits a needle, a microneedle that encapsulates the drug. And we've tested a dozen molecules, done thousands of deployments at this point, hundreds in clinical settings in humans, over 100, I should say. And it's proven to be reliable and effective. You know, we still have a lot more clinical work to reach like an FDA approval and demonstrate that conclusively. But in our early work, we have a lot of confidence based on the performance of the device and the bioavailability we're getting from our clinical studies.
Right, because that's always a big issue with things like vitamins and supplements, is that you get them into the body, but you have no idea how much is actually absorbed.
It's a great point. Things like iron are so poorly absorbed. Calcium has to be in certain forms or you don't get absorption. And we've actually thought about putting them into a needle and injecting them, and you would get much better absorption. The problem is the cost. Vitamins are pennies on the dollar. And so adding a technology to deliver them is probably not feasible. But for high value biologics, where you really want to expand the number of patients who are on it and make sure these therapies are being used and are efficacious, this is where an oral therapy could really be transformative.
The way that you were describing it, how do you guarantee or how do you ensure that it goes exactly where you want it to for when the inflatable balloon, for want of a better term, how do you make sure that it's in exactly the right spot?
So the target area is several feet. And the way we've designed this is the enteric coating, that pH sensitive coating that I mentioned that's on the capsule, is designed from how we tailor it, from the thickness, the materials we put in, to be very protective in the acidic environment of the stomach. And then in a very controllable and predictable way, once it reaches the small intestines, for it to dissolve and allow the mechanism to activate. So that's basically the timing of that is how we can ensure that it's in the right place when it delivers.
All right. And you said you've got like several feet of leeway as to where, okay.
And anywhere along the circumference, right? Doesn't matter the orientation either. So there's a huge area that's our target. I think we did the calculation and we assumed that, or we concluded that in any one centimeter square spot in that area, you wouldn't get more than an injection every year and a half or something like that.
And how often would that need to be taken? I guess that would depend on what drug you're delivering.
It depends on what drug you're delivering, absolutely. But what we would like to shoot for with most applications is moving to a daily administration. The reason being is patients, there are adherence issues with oral therapies, there are adherence issues with injectable therapies. Now, as I was talking about, the penalty for non-adherence with an injectable is much higher because typically the period between the injections is longer. So you could be at a really low trough by the time you need your next injection. And if you miss it by a week or you skip it altogether, you might be completely out of therapy for a month. But if you can move to a daily dose administration, particularly with monoclonal antibodies that have long half-lives, you can't make patients completely adherent. That's not a reasonable or realistic goal to get to, but you can eliminate or reduce and minimize the effects of non-adherence. So that's the benefit of going to a daily administration. And people are used to, like you were talking about, take a vitamin every day. We think that that's something from our research as well, that there was no strong preference over taking a smaller number of pills per week, one pill a week versus one pill a day.
It's quite complicated in terms of there's a lot going on there, but as you said, some of it gets excreted and the rest of it dissolves. So it's not like all of this is building up in your body.
No, nothing really remains. The injector portion of the technology is excreted out. The needle is made of sugar or another dissolvable polymer. It disappears in 10 minutes or less. And the drug is the drug. The drug is taken up thereafter. So from our perspective, relatively benign. It's an injection, just like an injection in your arm, essentially, and then everything else goes away.
And in terms of producing this at scale, is it cost effective?
Yeah, great question. So one of the reasons that I didn't describe electronics, microcontrollers, springs and levers and metal components or gas cylinders or things like that, is they have very high COGS associated with them. And we're aware with our goal of getting to a daily dose administration that the cost of goods had to be as low as possible. So plastic, sugar, Alka-Seltzer as our inputs are de minimis. And so it's really just manufacturing, automation, scale, capital equipment, amortization, and people. Our targets when we're at a commercial scale are around about $1 per pill per day.
Yeah, that's definitely reasonable. What kind of conditions are you working on in terms of the ability to use this?
Well, this is what's so exciting about Rani Therapeutics. Biologics have proliferated over the last 30 years, 20 years in particular, and they're in virtually every chronic disease category. So anywhere where patients have to regularly inject themselves is a good candidate for a Rani pill. And so what we're working on right now are primarily endocrinology and immunology categories. So our RT102 program that we had a phase 1 and a repeat dose study this year that we shared data from is for the treatment of osteoporosis. So this is primarily postmenopausal women. Our next program, RT111, is an ustekinumab biosimilar sold under the brand name Stelara by Janssen. It is used for psoriasis. And then down the line, there's going to be applications in psoriatic arthritis, potentially rheumatoid arthritis, some rare disease categories like hypoparathyroidism, and we'll add more as we go. As I said, there's plenty of other places to go. Obesity is another really interesting one that we're looking into.
All right. You mentioned osteoporosis. Can you give me some of the results from that study?
Absolutely. We're really, really pleased with how that turned out. The drug that we're using, PTH 1 through 34, is an osteoanabolic drug. It's a bone growth drug. And it's one of the best drugs for bone growth in the category, but it isn't prescribed all that much because it has to be injected every day. You need a pulse. It's a hormone. So we believe that moving to a daily dose administration could dramatically increase the number of patients who could benefit from this drug, as opposed to what right now is mostly the standard of care, which is anti-resorptive. I call them the Rogaine of osteoporosis. Keep what you have, but you don't grow more bone. And that's great if you have mild osteoporosis or if you're using it prophylactically. But if you have moderate, severe osteoporosis, or God forbid, you have a fracture already, you really need to grow more bone. So that's the context for this. It's clinically dosed teriparatide, or PTH 1 through 34, at 20 micrograms. So that was the control in our study, a sub-Q dose of 20 micrograms. We tested 20 and 80 micrograms. The net was we showed lower adverse event rates, similar or lower, at 4 times the dose. So the 80 microgram dose actually had a lower AE rate than the sub-Q. And then on the bioavailability front, we showed 300 to 400% higher bioavailability than the subcutaneous injection. This is unprecedented, historic, whatever you want to call it, compared to all of the attempts at oral biotherapeutics that preceded us. Like I said, those were 1% or less of the current therapy. We're on the other side of that, 3 or 4 times higher. We also did a repeat dose study, which we released the data from a couple of weeks ago. And this was our first repeat dose experience in humans. We were able to show that the Rani pill was generally well tolerated in that 7-day, 10-subject study for repeated use. We showed consistently high reliability of the device. And we put in some early effort towards food effect. One of the questions we get, we were talking about vitamins again. Do you take this with food? Do you take it without food? Is there an effect on the performance of the technology or the absorption of the drug? What we found in our early work here, and there's more to do, was that there were as many successes after the subjects were given food as there were before, which indicates that food had no impact on the technology, which isn't surprising. This is a device platform. So unless the food is getting in the way of the balloon, it shouldn't make any difference, or if it was degrading the coating too early. But we didn't see any indication of that. We'll run a full-on food effect study in the future. But yeah, that gives you some color on our RT102 program. I guess the last thing I would add is that going forward, we have a pre-IND meeting, and our plans are to take this into a phase 2 in the second half of 2023. So we're very excited about this program and moving it forward.
Are there any other applications for this kind of technology? I could imagine it would be useful in veterinary field as well.
Yeah, absolutely. There are some monoclonal antibodies that are dosed to dogs. In particular, they have to be willing and able to swallow a pill and of the right size. So I think in the veterinary space, canines would be the best place to go. They'll readily swallow a pill. Pigs will bite your fingers off. Cats are too small. So I think that's probably the one where you could potentially use this technology.
Is this something that other companies are working on, anything similar, or are you in a position of uniqueness here?
I think based on the data, I'll answer it this way. We've pioneered this space. So we were the first to ever think of a swallowable auto-injector injecting anywhere along the gastrointestinal tract. And we have over 200 issued or allowed patents at this point to back that up, both on the fundamental technology, the combination with specific drugs, and manufacturing techniques. Now, we've demonstrated some success here in creating a step change in the absorption of these drugs and the broadness of the applicability, not just small peptides, but monoclonal antibodies and proteins. So there have been some large and small pharma companies that have been inspired by us and are trying, and we wish them well. We are going to continue moving forward. We're getting into phase 2s. We're unaware of anybody else who's even close. And then when they go to commercial, that's when we'll vigorously protect and exercise our patents.
What's the timeline from here in terms of, I guess, when we ultimately go to the pharmacy or the physician and get this prescribed?
Well, we can't wait for that to happen. And part of the strategy here is to accelerate that as much as possible. So we have strategically decided and chosen the drugs that I was mentioning. These are drugs that are already approved. So it's mostly a route of administration and perhaps a dosing reduction in terms of the changes. So with the RT102 program, we hope and are planning for this to be a 505(b)(2). That would allow us to use some of the historical safety data. Some of it will be predicated on how the phase 2 comes out. But that could mean that by 2027, we could potentially have our first product in market, not 10 years from now. And then thereafter, with the pipeline that we have and the drugs that we'll add over time, we could expect kind of regular approvals year after year thereafter, and bringing these products back into the formulary and to the pharmacy, but now in an oral formulation.
Is this US or is this going to be global?
We'd like to make this global. I think US, Europe, Japan for certain, but ultimately the patients are everywhere. And if we can bring this and improve people's lives, we will.
And I guess there's also a sustainability aspect to it as well, because needles have to be disposed of in a certain way. And
I'm so glad you asked that. We are actually going to do some work on this. We're putting together a white paper on this very subject. I found one statistic. I think there were 4 or 5 billion sharps back in 2006. That doesn't include like the billions we're creating because of COVID or because there's so many more injectable therapies. And these are biohazards. There's a lot of metal and plastic. They end up in landfills. I think this is something that we are going to look at the environmental impact of that. With a Rani capsule or a Rani pill, there are no more sharps.
Yeah, it sounds like a win-win for everybody. And I'm one of those people you were talking about earlier that absolutely hates injections. So
from our research, no one likes them. I shouldn't say that. There's maybe 10% of the population, 15% that says, okay, this is more convenient for me. I'd rather take it once a month and are highly compliant. But the overwhelming majority would rather take a pill every day. And the thing that I always say when people question that is, you don't go to the pharmacy, to the OTC part of the pharmacy and buy injectable ibuprofen. No one in their right mind would develop that, because if there was an oral formulation already on the market, there's no benefit or upside to the injectables.
All right, those are all the questions I had. Is there anything else that you wanted to cover that we didn't?
No, this was great. I just want to say thank you so much for the time. Appreciate being able to share the story. As you can tell, we're really, really enthusiastic and excited about what we're doing, and can't wait to get into 2023 and get back into the clinic.
And that's it for another week, and another month, and another year. It's definitely flown by, and while I'm looking forward to the holidays and some relaxation, mainly because the forecast is for a week of rain, I'm also looking forward to next year, to meeting, online and in person, more people with more technologies and more research that's making a positive difference in people's lives, which I guess is really what it's all about. My first trip of the year is also coming up in less than 3 weeks, when I'll be popping over for a lightning visit to the capital of Northern Ireland, Belfast. Of course, before that, there's a certain mountain of wrapping paper to create and then to recycle, Christmas music to be listened to, some good and some far from it, and no doubt some overeating to participate in. I see there's bad weather in the US and Canada, so if you are listening from there, safe travels. And may I wish all of our listeners a fantastic holiday season, whether you're visiting family, relaxing at home, being woken at 5 a.m. because Santa has arrived, or just getting away from it all. And of course, because the next podcast is in January, I also wish you all a happy, safe, healthy and prosperous new year. So I hope you enjoyed this week's shorter podcast, and that wherever in the world you may be, you have a great couple of weeks ahead, and you'll join us in January for another Beyond Biotech.